recording this. So my independent variable in this case, you could compare temperature if I'm going to use temperature as a variable, but if I'm going to do that, I need to compare germinated seeds for 1 day. That's my experimental group. 1 day old seeds against 1 day old seeds. And then I'm also going to I could be doing this with germinated seeds, but if I'm going to be comparing germinated seeds that are 1 day old or 3 days old, I need to be comparing them at the same temperature so I have my controlled variables handled. What do you guys think is the real control group in this experiment? Let's go to that person. Let's go to George. I'm sorry to give you an easy one, George. What'd you say, baby? Did you say something?
I was saying, could you repeat the question?
Yeah, what do you think the control group is? It's right here. Which is the control of these three? You got to have a control group. Which is it of these three groups?
Would it be the glass beads?
Yeah, the glass beads. They're not living organisms, but they do have the same shape, right? They're little beads, so they have the same shape as the seeds. They probably have the same number of them in there because we're measuring volume. So the displacement of the glass beads should be equal to the displacement of the seeds as well. Which group had the greatest increase? We already talked about that. That was going to be the group that's 3 days old. So they're older, they're bigger. And I think I think that this is a pretty nice, data table for them to ask a lot of questions for. I went on the ACT. These were essentially the questions, but they had multiple choice answers, and I took away the multiple choice answers. There's one thing about experimental design and doing the labs. It's going to help you guys on the ACT because you understand how these things are set up and understand how to interpret data tables. Now I'm going to go to the next part. We're 2 minutes behind today. I need to get back to the review. So we need to review before we take the quiz. There's going to be a quiz in about 10 minutes. So let's get through that. So I'm going to go back and I'm going to look at respiration, but this time we're going to looking at anaerobic respiration. This little black dotted line, this is going to essentially be glycolysis. And so I want to...
Mr. Pope, we can't see your screen.
Oh, thank you. Thank you so much. So this dotted line here, this is indicating glycolysis. And so while it's technically, this is not fermentation, this is glycolysis. And this blue line is going to be fermentation. And I wasn't very clear on that. I went back and I looked at the video that I from the last class, and I thought, oh man, I really didn't do a good job with that. This is technically glycolysis, and sometimes we'll kind of lump these together. But let's just stick with this particular part of it, which is fermentation. So my product, and in this case, pyruvate, 2-NADP, and 2-ATP, this is the products of glycolysis. But my pyruvate, which was, came from the glucose, and it has been now oxidized, and we've got to recycle this NADH. Since there's no oxygen for this to go to the electron transport chain or oxidative phosphorylation, I've got to regenerate it. I have to have NAD to come here and break down glucose, to strip electrons off. It's oxidized and has to be reduced. And when I oxidize glucose, I'm going to get pyruvate. I'm going to break that. I'm going to oxidize glucose to reduce NAD into NADH. And I generate pyruvate. But I've got to get NAD back to keep this process going. Again, this is just fermentation. This is respiration without oxygen. This is anaerobic respiration. Now, this is what we use. Now, microorganisms, whoa, what happened to my, oh, I don't have alcoholic fermentation anymore. That sucks. Alcoholic fermentation is essentially the same thing except you produce CO2, but I went over that last Where does, okay, so Monas, where does alcoholic fermentation take place? It's a good question. So let me ask you a question. And I'm going to stick with you on this, Monas. Let's make a logical, let's make some conclusions based on information we already have. Let's deduce an answer. Where does glycolysis take place, Monas?
It takes place in the interior membrane space.
I don't think so.
Am I thinking of the wrong?
I don't think so. In fact, let's go back and, man, I don't know why these aren't coming up. Man, my images are not coming up on my document anymore. I don't know why. This is kind of frightening me. Hold on a second. There it is. I wonder why my image doesn't show up. Super weird. I see it here, but I don't see it whenever I get it. Don't know why that's the case. You can see it here. You can see the image here, but it's not letting me open it otherwise. Glycolysis does not take place there. Let's see if that one shows up. None of my images are showing up on this document. It's all right. We don't really have to have them. Can somebody help him out? Where does glycolysis take place?
Oh, it's the matrix, isn't it?
Glycolysis, I've got it right here in this data table. So let me go back and make sure that you can see this again. So glycolysis is occurring in the cytoplasm. Remember that this is anaerobic respiration, does not require mitochondria. Glycolysis takes place outside of the mitochondria. Glycolysis is going outside. The pyruvate diffuses into the mitochondria, is where citric acid cycle and electron transport chain are taking place. So if glycolysis is taking place in the cytoplasm, then fermentation is also taking place in the cytoplasm as well. All right, so let's go through this again. This is going to be individually, and last time as a group, the group that won last time got 5 extra points for the quiz. I'm going to go through this individually this time. We have got to go a little quicker today. So in this case, alcoholic fermentation, what are going to be the starting molecules or substrates for alcoholic fermentation? Knowing that fermentation is taking the products of glycolysis, and that might help you right here. What are the molecules that you need to start this process? And I'll go individually to you guys. And so Chastity, we're going to start with you. What would be the reactants for alcoholic fermentation?
Glucose, ADP, NAD+.
Okay, and for technically, yeah, if for glycolysis, I would include that. But what's coming out of glycolysis, and if I include glycolysis and alcoholic fermentation together, you would definitely be correct. I'm going to stick with the products that are coming out of glycolysis that we're going to use, and I did not do a good job last time in doing that. I lumped those two together. Technically, it's pyruvate and NADH. Honestly, you did get that correct, but that was both glycolysis and fermentation. So let's see, what are my products? What are the end products for fermentation, of alcoholic fermentation? Let's go to let's go to Weiss, Segal. What are the products of alcoholic fermentation? You have to take a stab at it to stay in class, otherwise I bounce you, kid. You've got to take a stab at it.
I don't know.
Oh, well, you could do that. There was one, it's ethanol, right? I know if NADH is going into it, it's going to produce NAD. I know this. This is a substrate. And then I'm going to produce alcohol and CO2. So if you see a substrate, you can most of the time predict a product. Let's go to the location. We've already discussed that. That was a cytoplasm. That's a give me. How many ATP are synthesized? How many ATP are synthesized? In this case, I'm going to include glycolysis in this. So if glycolysis is going to produce 2, then alcoholic fermentation is going to allow glycolysis to still produce 2. But these aren't directly from fermentation. They are from glycolysis. I want to make sure that you get the point that it's NADH being recycled to NAD plus that allows that process to continue. What are the electron donors and acceptors involved? And you don't have, all you have to do is look over here to see those. All you have to do is look over here to see these. So let's go to Dev. What are the donors and electron donors and acceptors?
NADH
And
and FADH.
I don't think so. I don't see them involved. Look, you can see over here.
Oh wait, no, NADH and I'm not sure what the second one would be.
NAD itself. NAD
Oh,
itself.
okay.
And
Okay.
NADH is the reduced form. That's why I have it in bold. It's reduced. Lactic acid fermentation is exactly the same thing. It's all about recycling NAD. And my products are a little different. We just saw those. So let's go over to LeVar. LeVar, what are my products? The products are going to be a little different. Buckman.
Is it glucose? I mean,
So you're saying we're going backwards? Because we got pyruvate as a substrate.
is it NAD plus then and FA?
Nope, no FADH. That's only going to be occurring between the Krebs cycle and electron transport chain. Look here. This is alcoholic fermentation, and we get alcohol. If this is lactic acid fermentation, what do you think we're producing here?
Lactic acid.
There you go. And lactic acid.
Oh.
That's right. Yeah, it's not too bad. So cytoplasm as well. That's an easy one. 2, that's easy as well. And then my electron donors and acceptors are going to be the same. There's very little difference between those. All right, I'm going to release a quiz. You guys are going to have 6 minutes for it, and then we'll reconvene. I'm going to go to Schoology, go to today's lesson, refresh your page, and you will see a quiz. You've got 6 minutes. Once you start taking it, I will start teaching again in 6 minutes. If you've just joined us, which some people have, the quiz is open. There it is. There's only 4 questions, and I will start teaching again in 6 minutes. Now 5 minutes. Actually, You guys have 1 minute. And again, I'll post a second version of that quiz. If you don't like that grade, I will post a second version of it that you can take this afternoon. All right, that was 6 minutes. All right, so I'm going to explain to you guys this is a really a traditional simple lab setup. You can see again, this is the, P's are a classic example that people use for respiration. And keep in mind that you do have your IA next year and how you want and what lab that you want to use for your IA could be determined by what resources you have. This one's simple, and this one we do have the resources for. In fact, I've got a lot better materials in this. This is more of a classic example that is often used on IB exams. In fact, you're going to see it. You'll see a version of this on your exam next week. It's really straightforward too. So I've got my P's. This is my experimental group. My glass beads obviously are going to be my control group. I have some materials. I don't know exactly what the independent variable is here. This is just the setup. My independent variable could be temperature, because temperature is a variable with respiration. It could be that they have another group of peas that are germinated at 4, 5, 6 days, and these are 2 days. I'm not sure. That's not really the point. Let's just talk about what these resources are and how they work. So I have an enclosed chamber. I've got a test tube. This is my stopper. I've got capillary tubing. In this capillary tubing, this little black line or little section here, this tubing, could be either oil or it could be water. And what it'll do is pressure falls in this tube. This little dot or this little bead of oil or water will migrate through the capillary tubing as it consumes oxygen. But there's a problem. When you consume oxygen, you're also producing CO2. So we've got to make sure that we eliminate the carbon dioxide in the tube so that we can measure the oxygen fall. If there's carbon dioxide that's being produced while oxygen is being consumed, then they're going to cancel each other out. So what we have to do is we have to get rid of the carbon dioxide. This is potassium hydroxide. This is KOH, potassium hydroxide. And what it does is it reacts with CO2 and turns CO2 from a gas to a solid. Basically, this is Drano. And honestly, that is what Drano is potassium hydroxide. That's all it is. So they put cotton balls above the seeds to protect them from the potassium hydroxide, because obviously Drano would kill peas. And you put that in there, and as they produce carbon dioxide, it turns into a solid. It reacts with the potassium hydroxide that CO2 does, and it cancels that out. And you watch this bead drop, this little bead fall, and you'll see the bead fall here as well. Maybe it wouldn't be because of respiration, but it could be just slight, subtle changes in the air pressure in the room. So sometimes you're going to see a little bit of a difference in both of these capillary tubing, even though that these glass beads obviously are not organisms consuming oxygen. You'll just see differences in the barometric pressure for the day, like we've got a cold front coming through today. Actually, barometric pressure is actually decreasing today, so that little bead might push this other way. But you could do stats to see if that's due to just chance, or if it's actually going to be respiration. The rubber tubing here, this little tube here on top of this, is going to cut off the oxygen, seal it closed. So there's a seal here, a seal here. This is completely sealed off. And this is just the airspace that has the oxygen in it for them to actually use. So let's look at another design. What do you, there's 3 designs here, and I want you guys to tell me, what are these labels for? So what do you think soda lime would be for on these experimental designs? So let's go to Sarah Marino first. So what do you think the soda lime? We've got a grasshopper, we've got cotton wool, just like we had for cotton. What do you think the soda lime is going to do? Sarah, I need you to give me an answer. Or I'm assuming that you're not there, Sarah. Sarah? No. Yeah, I'll repeat it one more time. I mean, right now we're running behind. I need you to focus. Soda lime, what do you think this resource is for? Okay. What do you think this is for, the soda lime? Okay, so let's see if somebody can help you. She says, I don't know. So, Monas, what do you think the soda lime is for? We're measuring oxygen consumption. What that little, so what do you think it would be? The soda lime wouldn't be like a filter. The cotton wool would be a filter. This is going to be, oh, hold on, there's 4 people coming in the chat. All right, go ahead, Daniel. You might know. Go for it, baby.
I'm thinking it's for like a closed environment. Like it's like a type of acid or something.
It would, it's going to be, so if CO2 is going to be produced, remember that in respiration, you produce CO2. If we're going to measure this droplet migrating down this tube, we've got to make, and that's going to be because oxygen is being consumed. So this little, air pressure is going to fall. It's going to pull oxygen out of the environment. We've got to get rid of the CO2. So the soda lime is going to be to get rid of the CO2. The cotton wool is going to be to protect the cricket. Yeah, that's going to be to protect the cricket. So we don't want the cricket to die. We want to make sure that the cricket stays alive for 2 reasons. We want to be ethical and keep our animals safe, but we also want to make sure it's alive so we can actually measure respiration at all. What the water drop is going to be measuring the fall in oxygen. They're going to be breathing, and as they're breathing, that little water droplet is going to move through here. So in that case, the meniscus here is going to be doing the same thing. The meniscus is going to be migrating to also measure oxygen falling. It's going to be measuring oxygen being consumed. What do you guys think the sodium hydroxide solution would be used for? Let's try somebody else here. So Kate, what do you think the sodium hydroxide is going to be used for? This is a different experiment here. These are going to be my germinated seeds.
So does this one have like a similar setup that it's meant to measure, or it's meant to counteract the carbon dioxide production?
You're correct, ma'am. You are correct.
Oh, really?
Yep,
Nice.
we've always got to get rid of that CO2. I can't measure, because remember, O2 is going to be, in respiration, is going to be increasing, and CO2 is going to be, I think I said increasing, decreasing O2, and CO2 is going to be increasing. So in order, these would just stay balanced. This meniscus would never move. This little water droplet would never move. So we've got to get rid of CO2. All right, let's go to, I've got more people. All right, so the screw clip. Why do we think we have a screw clip here? So let's go to Becerra. What do you think this little clip here does?
Holds everything together,
Everything together, so it holds
like the tube together, because it's a rubber tube, so it holds it together.
Well, this whole thing's a rubber tube. This whole thing's a rubber tube. What do you think it's doing at the top of the rubber tube?
Closing it for
Yeah.
outside.
Yes, sir. That's right. It's going to be a seal. So it's going to be a seal. I want to make sure that my environment is going to be sealed. But the meniscus, what is the meniscus measuring? So let's try my good friend. I have good friends in this class. Yang, what is the meniscus measuring? Did it over here. What is the meniscus measuring? O2, is it consumption or production? Gang, you got to take a chance on it or I'm going to bounce. We're going to bounce.
I'm not sure.
Oh, well, in respiration, do you produce oxygen or do you consume oxygen?
Produce?
So you produce oxygen, so I can go into your room and you produce oxygen like a plant?
Create.
Yeah, you're going to create oxygen? Or do you inhale oxygen?
Oh, yeah, I take in oxygen.
You take in oxygen, yeah. So that's going to be the decrease in oxygen. Oxygen being consumed, that's what that's going to be. So pondweed, what is this going to be? This is going to be where over here I had seeds. Over here I had a grasshopper. What is my pondweed going to represent? And let's go to our beautiful friend, Carlos Alvarez.
I'm assuming it has to do something with the carbon dioxide.
It's going to produce carbon dioxide, and that carbon dioxide is going to react with the hydrogen carbonate, basically baking soda here. But what? Keep looking. Just tell. I'm writing out the word. I'm hoping that you'll.
Organism?
Yeah, it's the organism. Yay! Now this is pondweed, so it's a plant. So do you think this plant is in the light or in the dark to be measuring respiration?
In the light.
So why do you say the light?
Because it's inhaling oxygen.
So in photosynthesis inhales oxygen?
Or it consumes.
It consumes oxygen. So do you need light for respiration to occur? If I turn out the light, so if I turn out the lights, you suffocate. So you keep the lights on in your room 24 hours a day. You don't suffocate when you turn out the lights?
No, not at all.
Okay, you're not afraid of the dark. I'm me either. I'm not afraid about it. Yeah, so this, in order, if I were in the light, so let's say that there was light for a moment. Let's say that we were in the light. In the light, you're going to have photosynthesis and you're going to have respiration occurring, both. But if it's in the dark, what is the only process? And I'm going to stick with you, Carlos. If it's in the dark and I'm a plant, what is the only process occurring? In the light, photosynthesis and respiration occur in the light. But in the dark, what is the only process that's occurring?
Isn't it the same thing?
So photosynthesis doesn't need light. So plants, if if we lost the sun, they could still use the photosynthesis?
I mean, it's not gonna change, but if it's the sun,
Oh, I'm
I'm
saying the
assuming
sun's down. It's at nighttime. Or the plant's photosynthesizing at night.
no.
No, I mean, that's the whole word, photosynthesis, light synthesis. So in the dark, which is the only two of these processes that is going to be occurring?
Respiration.
Respiration, right. So this lab setup here would be only occurring, if you're going to do this, you would only be able to do it in the dark. That would be the only time that you could do this particular one. So here, let's look how temperature works. So I've got temperature. I've got germinating corn, and that germination is at 22 degrees and 12 degrees. So which one has a greater effect? Well, obviously you can see that this one has a greater effect. Over here, we're looking at consumed milliliter of oxygen consumed. At 20 minutes, we had almost probably about 1.75 is about right. Maybe not. No, that's 1.6 if we're right at 20 minutes, 1.6. We're at 12 degrees Celsius, it's going to be much lower. It's going to be 0.8, half of that. Non-germinated corn is going to be, looks like almost 0.1 and 0.2. I don't know how significant that is actually going to be. There could have just been some changes in, there could have literally been changes just in pressure in the room. You would have a control group that you would be measuring this against just to make sure. But it's not always going to be that straightforward. This is data that we generated 2 years ago. If you were in class, you would have used crickets and mice, and you would have run this experiment in class. I want you to notice something. When we run this line here is at what temperature? This top line. What is the temperature for this mouse? What is the temperature there? So let's see.
5 degrees Celsius.
Yeah, it's 5 degrees. So is that cold or is that warm? 5 degrees. That's about 40 degrees Fahrenheit. It's pretty chilly. And then we had them at room temperature, which is 20 degrees, and that's about 68 degrees. So at 20 degrees Celsius, they didn't use respiration so much. Remember, there is thermal energy release in respiration. That's what keeps you warm. Your body temperature is due to respiration. You breaking that glucose down generates some heat. There's some heat that's lost. But when you look at the mouse, I know it's hard to see here, but I mean, excuse me, not the mouse, but the crickets. You can see the crickets are actually, it's the opposite. Their respiration was higher at 20 degrees and lower at 5 degrees. It was actually lower. But there's a difference between a mouse and a cricket. What is the difference? Well, one is an endotherm and one is an ectotherm. So an endotherm is it means this is inside and ecto is outside. Another way of saying this would be endotherms are warm-blooded, and that means that would be the mouse. They're warm-blooded. That's my mouse. But what if a mouse is warm-blooded, and that's what endotherm means? What do you think ectotherm means? Then it's going to be cold-blooded. So if we were doing this in the classroom, which we would, these crickets, their body temperature is dependent upon whatever the temperature is in the room. If you get cold, you have to produce respiration. You have to use respiration more to make sure that you maintain a body temperature. Heat is going to escape from you, and you have to burn more calories to stay warm. So you guys want to lose weight? Go sit your butt outside in the cold yard with no clothes on, and you'll lose weight faster than if you sit out there in a warm yard, especially if you're exercising. You've got to replace heat that is lost from your body. And that is going to be due to this situation. Now, I had some data to play with, but there's no way. We didn't get to that. So I'm going to end right here, but I do want to show you one thing, and I know I'm taking up 1 extra minute of your time. And this is going to benefit you to do this is the last time that we meet. There are different data sets here. Your data is not organized very easily here. It's not organized very easily at all. And well, I'll say easily organized. It's not well organized, would be a better way of saying this. This data isn't organized in a way that you can use in a data classroom. What would we have to do to organize this data? What would we have to do? If you come in here and you look at this, you remember you have to have your independent variable in one column and your dependent variable in another column. So what you'd have to do is take this, you got temperatures here. You take your temperatures and put them over here, because these are all ectotherms of keeping a constant. I put my temperatures on one side, and I would put my dependent variable of carbon dioxide consumption on the other. That's what you would need to do. So what I would do, instead of changing this to thermi, I would just make this temperature, and I'd make this 6 degrees Celsius, change that all the way down here. Oh, it changed it to 15, but I would change all these to 6. I would make all these 6 degrees and all of these 22 degrees, and then I would take this data and I would copy it and paste it here. And if I were you, and I am not, but if I really wanted to do well, I would go through here and I would graph this data on Data Classroom and see how endotherms and ectotherms are different. And I would change these to 22 degrees. Change all these to 22 all the way down. And I would graph this data on my own. I have to let you guys go to the next class. You guys be good. I'm going to stay here and do this so that you guys can see it. Those of you guys that want to stay, or you may not stay, but that want to watch the recording, I'll let you guys watch the recording. And if you are my third period, you want to stay here and watch this recording because you get an opportunity for something. I'll let you know what it is if you stay long enough to watch the end of it. See what my chats are. Got some kids chatting. Nope, nothing's due. You can take the, if you would like to take the retake for the quiz, I'll post it in about probably 30 minutes, but I'd take it after all my classes were over. Okay, so I'm going to get rid of this column now. And if you're a third period student, you need to do this. This is something that you're going to need to do. So I'm going to put my units up here. These are my units. I've got a Celsius is my units. I'm going to save this file. Say, maybe I won't necessarily. Oh, I got it in a Google Doc. So let me save, make a copy. Make a copy. Am I copy? Copy Ecto 1, 1. So it doesn't script your other copies. Now I'm going to go to Data Classroom. Make sure that you guys can see this. And you guys that are still here, you do have another class to go to. If you want to stay, you're welcome to. We'll go to Data Classroom.
Goodbye.
Goodbye, kiddo.
Hi, Mr. Poe.
All right, be good.
Bye.
All right, yeah, goodbye. I'm just recording this for third period. When you go to Data Classroom, you're going to sign in with Google. You should have an account. I'm already synced there. And then I'm going to import this data, this data set, and I'm going to fetch it. I'm going to fetch this data. So let me fetch this data from my Google Drive. I'm going to put the link from my spreadsheet. It's right here at the top. There's a link to it. So I'm going to go to my spreadsheet, copy this link. I'm going to put it in here and fetch. Oh, Google login required. Sorry. Now it works. Done. Okay, so now I have, make sure I'm still sharing this with you guys. I'm gonna make sure I'm still sharing my screen so it didn't jump away from you guys. And we're doing ectotherm. So here, I've got all my data. I need to change this data type to categorical and this data type to numeric because this is my independent variable. Let me get my picture out of the way here, stick it up here. And I'm going to make a graph with it. So I'm going to show, make it a scatter plot. And for some reason it's not showing up. Oh, got to show a Y and X. So I went over here and I changed Y and X. Now I want to know if this is significant or not. Oh, there's somebody chatting with me still there. Bye, Mr. Yeah, bye, George. So I'm going to put descriptive statistics. I want to change standard deviation to standard error, and they're significantly different. If I want to get my p-value, I'll come over here to get my p-value. So there's a little magnifying glass with a p in it, and this is set up for a t-test. I will calculate this and see what my p-value is. It's 0.01, so it is significant. And this was data that was generated by kids in the class a couple of years ago. Now, what you guys have to do for me and if you do this today, and I can go in here and I'll show you what I mean, how I know that you've actually done this or not. So if I go to if you do this today, you build a graph with this data, you're going to get 5 extra points on the quiz today. And you guys do have a retake as well. I'm talking the third period. So what I would do, I'll go to my classes. I'm not going to, I'll discard my changes. It doesn't matter. If I go to my classes and I look at my students, and you guys are third period, show members, I can come in here and I can click on, let's say Aspen, Jenkins, Romello, Abby, whomever, and I can see, did you do it or did you get a picture from somebody else? It's not hard for me to actually go in there and do that. So if I go to users, like I can see Aspen Jenkins, he was on here 3 months ago. And if I want to click over here and check out what Aspen did, I can see what his actions were. So in order for you to get that extra credit for third period, get the 5 extra points, what you've got to do for your quiz today is build me a graph. And when you build the graph, just like I did, and you send it to me, I'll come in here and I'll check and see if you did it or somebody else did it. And if you did it, you'll get those extra points. But hold on, I'm going to show you one other thing that you need to do. This is just for 5 extra points. Here's our class for today. Wrong period, your third period. Everybody has their own data set. For your class is a little different than theirs. Everybody has their own data set. You come here, this is the sample data that we started with. You can choose endotherm or ectotherm, and I don't care which one. You've got to change the data in here to fit the way that it needs to be. Just like I organized it, you've got to organize temperature and CO2 production. You've got to change the format and save it so that I can go back and I can check it. You'll have to save it in order to run it on Data Classroom. And if you need some help, I'm going to be at the doctor. I cannot talk to you today unless you catch me after school, but I have to go to a hospital and I will not be able to be talked to or communicated with probably until about noon. If you call me between, say, noon and 2, I'll be available to talk to you. My number is 817-714-2886. All right, you guys be good.